Okay, hello, good evening, everyone. Yeah, I see that this is maybe the second generative machine learning talk, and in the previous one, it was house full with people on the stairs. I hope I'm able to make it as exciting as Mo did for the rest of you that remain. But today we'll be talking a bit about essentially multimodal generative machine learning for non-clinical safety as a process. I will take a few seconds to describe what exactly I mean when I talk about sort of non-clinical safety and why it's kind of like a topic of my choice instead of, say, for example, talking about let's say more attractive topics like early discovery from a VC sense. So I'm sure like everything that I show here is not much of a surprise to people in the room. Like drug discovery is a challenging process. That's no news for us. This is what the numbers and the odds look like. And today we have a lot of hope because of a lot of automations that have come to fruition due to this amalgamation of data and compute in a perfect Goldilocks zone, at least for parts of the process that can reduce the timeline. So essentially, like, when you have early discovery, which you do a lot of the work in vitro, is followed immediately by a preclinical phase of analysis where substances identified during early discovery, they need to be essentially optimized and tested for safety and efficacy guarantees. Now here, of course, like there is a lot of discussion to be had about to what extent is animal model testing as a process as regards non-clinical safety useful for the overall big picture for drug discovery, and B, obviously this perennial question about the ethical dilemmas associated, given that there is a lot of like ex vivo imaging that is involved that fundamentally requires sacrifice or euthanization of animal models ranging from murine to porcine to canine and so on and so forth. So today there is this other question that also gets asked very often, that given that there are kind of like ideas that essentially when you talk about something like drug safety evaluations and you talk about the role of animal models therein, there is a lot of like studies out there that talk about to what extent the utility of that translates into final approval rates, with different studies claiming about 72 to 90% of animal testing as being non-conclusive for eventual success in clinical trials. So with these ideas in mind, eventually in 2022, there was kind of like a culmination of regulatory attention to this problem, with the FDA in its Modernization Act of talking about encouraging companies in pharmaceutical drug development and in allied processes to reconsider the need for animal testing and make active efforts in eliminating the need thereof. So the fundamental question there is, if not animal testing, then what? And to that extent, we propose that multimodal machine learning, multimodal generative machine learning can answer a part of that challenge. And as an exemplar of approach, I will today talk a little bit about toxicologic pathology as a process and to what extent we believe multimodal machine learning, particularly in the realm of foundation models, can help with the process in terms of exemplar downstream tasks that I will very briefly talk about, and will leave the field open for prospective directions towards the integration of different bioinformatics workflows. So just for a brief primer, what is toxicologic pathology? So fundamentally, toxicology, science of poisons; pathology, science of disease or science of manifestations of disease. It is fundamentally the study of like manifestations of particular pathologically relevant effects at the tissue level when you are in this context dealing with animal models during the drug development process. So why do we talk about AI in that process today? It's fundamentally simply because of the large volume of slides involved, large volume of very normal slides with essentially an out of distribution situation where you see toxicologically relevant lesions. Now these numbers are actually not very small, as you know, a lot of you in the room, that a single IND can involve anywhere between like 80 to 100 animals and about 60 to 80 tissues per animal model. So essentially we're looking for a very time consuming and labor intensive part of the process. So even without any of the other concerns or regulatory encouragement, there is a very strong case of inherently building automation into this part of the drug discovery and development pipeline. So we do know, for example, like you have a lot of these studies over the years that say that there is such a large volume of slides pathologists exploited in the industry. Like you have this number from 2019, which is actually quite dated now, from Charles River. They're sitting over 4 million microscopic slides. It is fascinating to see that this is one of those industries where the perception has moved so fast from essentially sort of pathology scanning for animal testing being a process that was outsourced, with the slides essentially being byproducts that were not particularly useful afterwards, to today being a gold mine of resources to actually building digital algorithms to help with the part of the process. So a lot of that obviously has been enabled by the improvements in the tool chains that we have and the emergence of bespoke algorithms to handle the particularly data rich and also at the same time data poor in a very quality sense in toxicologic pathology. So you do have kind of like a bit of prior work today, but this is a field that is kind of burgeoning still. And in one of the reviews that I do not mention here because it is a part of a peer-reviewed publication now, we concluded that there were precisely 77 different papers that dealt with some kind of machine learning for toxicologic pathology that exists in literature today. So not a very large number, but still an evidence of growing interest. So as such, the idea is that if we are talking about like essentially the presence of data, the presence of a business case, and the presence of compute, what are those key downstream applications where we get our wet lab and pathology teams most excited about? And one of the core ones is understanding the liver as an organ, or arguably the most vital organ for any organism when you talk about toxicity. So if you were to talk about, say, for example, processes that are routinely assessed with regards to evaluating the safety profile or efficacy profile of an investigational new drug, you would be looking at detecting tissue level manifestations of necrosis and of like identifying if there are mitotic figure enhancements or any kind of like calculation or any kind of like dramatic alterations to cell sizes. And thereafter, each of these can be translated into machine learning pipelines of different sorts, ranging from detection to segmentation to your cell counting applications and so on and so forth, all of which can be transferred potentially to other organs with appropriate amounts of fine-tuning and continual learning. Now, obviously, the question that also gets asked is that, of course, like clinical pathology as a field has existed for decades now, but digital pathology as a field is very novel, maybe not for us in the room who hear about that phrase all the time, but you talk to mainstream audiences in computer vision, the phrase is actually still quite new. But the question that does get asked in a more aware audience is that to what extent can you just, lift algorithms from digital pathology and put into toxicologic pathology? Now, the truism here is that at the end of the day, you look at an H&E slide, for a computer, it's ones and zeros. That itself, the morphology itself is not dramatically different maybe. But the core question is, when you look at clinical pathology as a process, you are nowhere close to dealing with a situation of, high kind of like, or you do not have the kind of signal to noise ratios that you have in clinical pathology. That simply doesn't exist in ToxPath. You simply do not have as many slides that have that diversity of pathologically relevant findings. There is substantially a lower need for the kind of applications that you have in terms of correlations to other modalities and of co-registrations with other modalities that are hyperspectral in nature. Those things are unique to toxicologic pathology as a domain, which kind of begs the question that we today have a situation where we know that there is a need for bespoke AI development for toxicologic pathology as a part of drug safety evaluations. If you were to look at a post-human or a post-animal model future for the same, then we do not have enough data that is out there. There is enough data, actually, to be honest. There is not enough labeled data out there. Then to what extent are we kind of like going to bring all of them together into that same kind of like, pot of soup that we are talking about? And the answer to that may be in the form of like self-supervised learning and representation learning from large-scale unannotated data that exists across CROs, across pharma companies, and across different veterinary institutions, not necessarily labeled, but substantially available in the form of slides to the point of learning those unique representations that define particular animal models. This is the same philosophical idea that underpins the concept of foundation models. Now, for a scientific audience, I must give a disclaimer. The phrase foundation models does not have a particularly scientifically robust definition, in my opinion. There was a Center for Foundation Models in Stanford that first came up with this phrase, but roughly it encapsulates the idea that if you have a large volume of data that is available in an open world scale, you can learn good representations by contriving tasks that allow for such representation learning in a composition of functions manner that a deep neural network kind of like represents. So essentially, those classic examples where you perform, for example, geometric transformations of images and get an algorithm to predict simply so that you can learn a representation of that underlying image space, and then you can, with much lesser high quality annotated data, fine tune those for particular applications down the line. That same principle can be adapted given the low annotation quality environment that we deal with ToxPath and eventually quite a bit of other healthcare applications. So today you have that idea that has been ported into different kinds of pathology use cases. You see that it would capture a broad spectrum of patterns ranging from morphology to staining characteristics and so on and so forth. And the kind of infrastructural development that we have in terms of the engineering stack being out there enables encouragement of going down the same route for toxicologic pathology. So obviously the field itself operates on a needle, discovering a needle in a haystack kind of a problem with the anatomically normal reality for a vast majority of the slides. So if you were to learn a very good representation of what a normal slide is, an out of distribution problem becomes sort of tractable for identifying pathologically relevant versions that of different kinds of pathologies across animal models that might be present for particular drug safety studies. So the question is, what is the way forward then? So like when you talk about the futuristic approach towards some of these things, we do know that regulatory toxicity studies, they generate a substantially large amount of multimodal data, including a lot of like omics data, a lot of like study reports and so on and so forth. So there is a value in kind of like looking at the foundation model problem from a more multimodal sense, where we look at kind of like combining multiple embeddings from across different modalities. And then, of course, we kind of like look into what kind of downstream tasks we might be best poised to answer. So the other question to ask, particularly in this context where drug discovery as a field has multiple cogs in the wheel, is that how foundational does your foundation model really need to be? Can we just stop at like, let's say, a slide level understanding of things and call it a pathology foundation model? Do we need to have specific foundation models for specific animal models? Or where do we actually kind of delineate the knowledge base of our foundation models really? So that is another question that definitely needs to be defined. And this might differ from candidate to candidate in my view. So going forward, I'll just very briefly touch upon some of the downstream tasks that we have shown interest in. So with this, yeah. Okay.
Can you go to the end? That's great. Thank you very much.
Okay, thank you.